Everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and subscribe the channel. In the last session, we ended up with metabolism of cardiac muscle. In today's session, we will start with effect of exercise on metabolic profile. First, it includes long distance running as well as weightlifting, which are typical examples of aerobic and anaerobic exercise. Then we will see how glycogenolysis changes to fatty acid oxidation. Then we will see metabolism in starvation, which include 5 stages, the last one being death. Then we will start a new chapter in which we will see digestion and clinical applications of carbohydrates. Good morning, everyone. Let us start the topic. Effect of exercise on metabolic profile. Long distance running is the typical example of aerobic exercise, whereas sprinting or weightlifting exemplifies anaerobic exercise. During anaerobic exercise, the major organ involved is the skeletal muscle, with very little involvement of other organs. The relative ischemia created by the compression of blood vessels in the muscle will necessitate the use of glycogen and phosphocreatinine available in the muscle to supply required energy. During moderate aerobic exercise, the muscular stores of glycogen are used, but in a normal individual, this is not sufficient to provide a continuous supply of ATP for exercise like long distance running. The respiratory quotient falls during long distance running since there is progressive change from glycogenolysis to fatty acid oxidation to meet the energy demands. Muscle starts oxidizing fatty acids and the high AMP level to activate AMP kinase and low malonyl CoA to activate CAD, which will favor fatty acid oxidation. The training of athletes is different depending on whether they are sprinters or long distance runners, since the energy sources are different. Rest after vigorous muscular activity often results in the depletion of the exhausted glycogen stores. In the muscle developed by exercise and training, the size and number of mitochondria are more, as well as the level of enzymes for fatty acid oxidation and ketone body utilization. Hence, the trained muscle can better utilize non-carbohydrate sources for energy. So, exhaustion is delayed. Coming to the next topic, metabolic adaptations during starvation. In early fasting, the effect of short-term regulation by altering the activity of existing enzymes, that is fine control, is more significant. When starvation is prolonged, that is more than 3 days, long-term adaptation sets in. For example, brain starts metabolizing ketone bodies, deriving about 30% of energy from ketone bodies. Coming to the first stage, glycogenolysis. During fasting, at first, blood glucose level is maintained by hepatic glycogenolysis. The glycogen stores are sufficient for about 18 hours. The primary requirement for glucose is to meet the demands of the brain. Coming to the second stage, gluconeogenesis. Even before the glycogen stores are depleted, gluconeogenesis is accelerated. The amino acids released from muscle form the major substrate for gluconeogenesis. The amino nitrogen is transferred from other amino acids to pyruvate and then to alanine. Thus, the amino group reaches the liver as alanine, where it is transmitted to give pyruvate for gluconeogenesis. This glucose-alanine cycle serves to transport the amino nitrogen of other amino acids to provide to a liver in a harmless form. Glutamic acid also serves as an important mode of transport of amino acids to the liver. In this diagram, we can see glucose-alanine cycle. Liver synthesizes glucose, which is transported to the muscle through blood. In the, mus- in the muscle, it is converted into pyruvate. Here it undergoes two steps. One is formation of lactate and the other is synthesis of alanine. Both are transferred back to liver and again glucose is synthesized. This is called glucose-alanine cycle. The branched chain amino acids liberated by muscle protein, especially leucine and isoleucine, are utilized by the muscle to give energy. Brain can preferentially take up the glucogenic valine from the bloodstream. The plasma level of branched chain amino acids reaches by the peak of fifth day of starvation. Coming to the third stage, lipolysis. The prevailing state of increased glucagon insulin ratio stimulates cyclic AMP mediated lipolysis by increasing hormone sensitive lipase. Then the skeletal muscle, heart, and kidney will shut down their glucose utilization and will depend on fatty acids and energy needs. Decreased pyruvate dehydrogenase by phosphorylation is the basis for this change. The stimulation of the activity by CAT by glucagon favors increased beta oxidation. The increased lipolysis and beta oxidation often provides an alternate source of fuel as acetyl CoA and subsequently ketone bodies. Ketone bodies provide fuel for the tissues like heart muscle, skeletal muscle, and to some extent to the brain. It is seen that brain starts utilizing ketone bodies from third day of starvation. By 10th day of starvation, about 60% of energy for brain is derived from ketone bodies. Coming to the fourth stage, acidosis. However, this state cannot continue indefinitely since metabolic production of ketone bodies is converted to metabolic acidosis. When the bicarbonate buffering capacity is exceeded, the pH falls and hyperventilation occurs as a compensatory mechanism. Coming to fifth stage, death from starvation. Metabolic acidosis and dehydration, unless corrected efficiently, will lead to death. A normal person has a fuel reserve to live up to 45 to 60 days. Examples are available in history. As a part of freedom struggle, Sri Jatin Das took fast unto death, who died on 61st day of hunger strike on 13th September 1929. Coming to next topic, major metabolic pathways of glucose. The digestion of carbohydrates. In the diet, carbohydrates are present as complex polysaccharides like starch and glycogen, and to a minor extent as disaccharides, sucrose and lactose. They are hydrolyzed into monosaccharide units in the gastrointestinal tract. Cooking makes the digestion process easier. This process of digestion starts in mouth by the salivary alpha amylase. However, the time available for the digestion in the mouth is limited because the gastric hydrochloric acid will inhibit the action of salivary amylase. In the pancreatic juice, another alpha amylase is available which will hydrolyze the alpha 1,4 glycosidic linkages randomly. It is because to produce smaller subunits like maltose, isomaltose, dextrins, and branched or unbranched oligosaccharides. The cells of fresh border of intestine contain the enzymes sucrase, maltase, isomaltase, and lactase. They hydrolyze the corresponding disaccharides to monosaccharides, which are then absorbed. Coming to the clinical application, lactose intolerance. Lactase hydrolyzes lactose, which gives glucose plus galactose. Lactase is present in the brush border of enterocytes. Deficiency of lactate leads to lactose intolerance. In this condition, lactose accumulates in the liver. Irritant diarrhea and flatulence are seen. There may be congenital, that is primary, or acquired, that is secondary causes. As age advances, lactase enzyme will be lost. Another reason for acquired lactose intolerance may be a sudden change into a milk-based diet. Lactase is an inducible enzyme. If milk is withdrawn temporarily, the diarrhea will be limited. Curd is also an effective treatment because the lactobacilli present in the curd contains the enzyme lactase. Lactate is abundantly seen in yeast, which could also be used in treatment. Coming to absorption of carbohydrates, only monosaccharides are absorbed by the intestine. Absorption rate is maximum for galactose, moderate for glucose, and minimum for fructose. Glucose has specific transporters which are transmembrane proteins. First, co-transport from lumen to intestinal cells. This process is mediated by sodium-dependent glucose transporter 1 or sGLUT1. Absorption from intestinal lumen into intestinal cells is by co-transport mechanism, that is secondary active transport. In this diagram, we can see sodium-dependent glucose transporter 1, where sodium as well as glucose are pushed against the cytosolic membrane. Coming to absorption of glucose, a membrane-bound carrier protein is involved which carries glucose along with sodium. This sodium is later expelled by the sodium pump with the utilization of energy. So energy is needed indirectly. The transporter in intestine is named as sGLUT1, whereas the transporter in kidney is called sGLUT2. The first one is involved in glucose galactose malabsorption, whereas the sGLUT2 is defective in congenital renal glycosuria. Coming to the clinical application, common treatment for diarrhea is oral rehydration fluid. It contains glucose and sodium. Presence of glucose in oral rehydration fluid also allows uptake of sodium to replenish body sodium chloride. Here ends our session. Kindly like, share and subscribe the channel. Thank you.